Choleragen. Choleragen, also known as choleragen and sometimes abbreviated to CTX. CTX or CT is a B5 multimeric protein complex secreted by the bacterium Vibrio cholerae. CTX is responsible for the massive, watery diarrhea characteristic of cholera infection. It is a member of the Heat-labile enterotoxin family. History. Choleragen was discovered in 1959 by Indian microbiologist Sam Punatha. Structure. The complete toxin is a hexamer made up of a single copy of the subunit part A enzymatic and 5 copies of the B subunit part B receptor binding, denoted as AB. Subunit B binds while subunit activates the G protein which activates adenylate cyclase. The three-dimensional structure of the toxin was determined using X-ray crystallography by Jain et al. In 1995. The 5 B subunits, each weighing 11 kDa, form a 5-membered ring. The subunit, which is 28 kDa, has 2 important segments. The AN portion of the chain CTAN is a globular enzyme payload that ADP ribosylates G proteins, while the ATWA chain CT-ATWA forms an extended alpha helix which sits snugly in the central pore of the B subunit ring. This structure is similar in shape, mechanism, and sequence to the Heat-labile enterotoxin secreted by some strains of the Escherichia coli bacterium. Pathogenesis. Choleragen acts by the following mechanism. First, the B subunit ring of the choleragen binds to G-mer and gangliosides on the surface of target cells. If a cell lacks G-mer, the toxin most likely binds to other types of glycans, such as Lewis Y and Lewis X, attached to proteins instead of lipids. Once bound, the entire toxin complex is endocytosed by the cell and the choleragen AN-CTAN chain is released by the reduction of a disulfide bridge. The endosome is moved to the Golgi apparatus, where the AN protein is recognized by the endoplasmic reticulum chaperone protein disulfide isomerase. The AN chain is then unfolded and delivered to the membrane, where Rune triggers the release of the AN protein by oxidation of protein disulfide isomerase complex. As the AN protein moves from ER into the cytoplasm by the Sexx-C1 channel, it refolds and avoids deactivation as a result of ubiquitination. CTAN is then free to bind with a human partner protein called ADP ribosylation factor 6-OFF6. Binding to R6 drives a change in the shape of CTAN, which exposes its active site and enables its catalytic activity. The CTAN fragment catalyzes ADP ribosylation of the GS-alpha subunit G-alpha-S proteins using NAD. The ADP ribosylation causes the G-alpha-S subunit to lose its catalytic activity of GTP hydrolysis into GDP plus PI, thus maintaining G-alpha-S in its activated state. Increased G-alpha-S activation leads to increased adenylate cyclase activity, which increases the intracellular concentration of 3,5-cyclic AMP-C-AMP to more than 100-fold over normal and overactivates cytosolic pKa. These active pKa then phosphorylate the cystic fibrosis transmembrane conductance regulator CFTR chloride channel proteins, which leads to ATP-mediated efflux of chloride ions and leads to secretion of H2O, NA-Ka, and H0- into the intestinal lumen. In addition, the entry of Na+ and consequently the entry of water into enterocytes are diminished. The combined effects result in rapid fluid loss from the intestine, up to 2 liters per hour, leading to severe dehydration and other factors associated with cholera, including rice water stool. The pertussis toxin, also an AB5 protein produced by Bordetella pertussis, acts in a similar manner with the exception that it ADP ribosylates the G-alpha I subunit, rendering it unable to inhibit CAMP production. Origin. The gene encoding the choleragen is introduced into V. coli by horizontal gene transfer. Virulent strains of V. coli hold a virus known as a CTX5 bacteriophage. Applications. Because the B subunit appears to be relatively non-toxic, researchers have found a number of applications for it in cell and molecular biology. It is routinely used as a neuronal tracer. Treatment of cultured rodent neural stem cells with choleragen induces changes in the localization of the transcription factor HESRI and increases their numbers. G-mer and gangliosides are found in lipid rafts on the cell surface. These subunit complexes labeled with fluorescent tags or subsequently targeted with antibodies can be used to identify rafts. Vaccine. There are currently 2 vaccines for cholera, Dukoral and Chancol. Both vaccines use whole-kill V. coli cells, however, Dukoral also contains recombinant choleragen beta RCTB. Some studies suggest that the inclusion of RCTB may improve vaccine efficacy in young children 2-10 and increase the duration of protection. This is countered by the cost of protecting and storing RCTB against degradation. Vaccine adjuvant. Another application of the CTB subunit may be as a vaccine adjuvant to other vaccines. It has been shown that coupling CTB and antigens improves the response of the vaccine. Currently, the adjuvant potential of CTB has been shown in large animal models, so further research is needed. This may allow for CTB to be used as an adjuvant for vaccinating against many kinds of diseases. This may include bacterial and viral infections, allergy, and diabetes. Of note, as CTB has shown to induce mucosal humoral immune responses, vaccines against mucosal viruses such as HIV are a potential target. Lipid RAFTS. Since choleragen has been shown to preferentially bind to G mononucleosides, this characteristic can be utilized for membrane studies. Lipid RAFTS are difficult to study as they vary in size and lifetime, as well being part of an extremely dynamic component of cells. Using choleragen beta as a marker, we can get a better understanding of the properties and functions of lipid RAFTS. Endocytosis. Endocytosis is broadly divided into clathrin-dependent and clathrin-independent process, and the choleragen utilizes both pathways. Choleragen has been shown to enter cells via endocytosis in multiple pathways. These pathways include CAVLE, clathrin-coated pits, clathrin-independent carriers, CLICs, and GPI-enriched endocytic compartments, GEECS pathway, ARS6-mediated endocytosis, and fast endothelin-mediated endocytosis FEM. How choleragen triggers these endocytosis pathways is not fully understood, but the fact that choleragen triggers these pathways shows the use of the toxin as an important marker to investigate these mechanisms. Retrograde trafficking. One of the most important aspects of choleragen is the retrograde traffic mechanism that transports the toxin from the cell membrane back to the trans-Golgi network in the endoplasmic reticulum. Since both choleragen and G-mon species can be tagged with fluorescent tags, the mechanism of retrograde traffic can be monitored. This opens up the potential to monitor the mechanism in real time. This may open up new discoveries on how intracellular transport works and how protein and lipid sorting work in the endocytotic pathway.